All right, muscles, part one, the second half of those flip notes. So, as I said, acetylcholine is very important. When the brain sends an action potential to a nerve, and the nerve's action potential travels all the way down to that neuromuscular junction where the nerve is touching that muscle, basically, it releases acetylcholine. Acetylcholine causes depolarization of the cell membrane, the muscle cell membrane, the sarcolemma. As soon as that acetylcholine is released, you got acetylcholinesterase, an enzyme which breaks down acetylcholine in that gap junction, attacking that acetylcholine so it doesn't continue to cause depolarization, so you can relax immediately. Like when you're sprinting, you can flex that muscle and relax it immediately to kick back into the next phase. So step by step, we go muscle contraction to know. First phase, once the cell membrane has been depolarized by the acetylcholine, calcium is released. It binds to troponin. Troponin is like the handle on the tropomyosin. The troponin then pulls the tropomyosin off the active binding site on actin. So now the binding site on actin is exposed. The myosin heads can grab a hold of actin. Once this occurs, ATP triggers the attraction. ATP is the energy molecule. It goes to ADP, and that causes the head to open up again. And this last part here, the ATP, when that myosin triggers and pulls, ADP is released, ATP pops on again, and that occurs several times in like a millisecond to make that contraction occur. So you got literally millions of these little sarcomeres contracting in your muscle fibers every time you flex any muscle in your body. So you're burning ATP just sitting there thinking, just sitting there with your heart beating. All right, this nice little diagram here, if you click on the dog's face there, it takes you to a link. You can see that link there, and it shows you this whole thing in action. And this diagram's nice. It shows the myosin heads binding the actin. What it fails to show is this band in the middle. Those actin myofilaments should come together and touch, which they don't, like right here. They should come all the way to touch. But basically, this shows you that you got 1, 2, 3, 4 sarcomeres demonstrated here by cartoon figure on the right. That shows you why you get striations in your skeletal muscle. Okay, if you want to get into more detail, this is what you see in college. This is what you have to learn in college. In high school, we don't go in that depth. Okay, again, you can see the sodium-potassium pump. Sodium and calcium is then released, which binds to the troponin, and you got a calcium pump right there in your smooth endoplasmic reticulum. So when a muscle cell is depolarized, again, the ions rush in momentarily. This happens in a split second. Okay, that's called the action potential. You've heard of action potential. And you heard that lady at SLU talk about how they spent a whole 2 lectures on action potential alone. We're not going to do that. All right, so muscle twitch is an all or none response. The entire sarcomere contracts or it doesn't. The nice thing about it is we control how much acetylcholine is released from each neuromuscular junction. So we can flex a little or a lot of these sarcomeres. Okay, again, there's the sarcomere. There are 3 phases of contraction. There's the lag phase, that little minute time between the stimulus to the start of the response. So when you put when your brain says flex, from the time the action potential is going down until it actually kicks in, lag phase. Contraction of the sarcomere. That's when the myosin heads actually bind and it starts contracting. And then relaxation of the sarcomere. It's got to relax again before it can contract again. All right, so contraction of muscle fiber in response, you have to have a threshold. Okay, you can think, get out of bed, get out of bed. The brain's telling your body to get out of bed, but unless you send enough information, you're not going to hit that threshold to actually trigger the action potential. So enough stimulus must be present to cause a reaction. Again, Saturday morning, they're telling you to wake up. Nothing happens until you get that cold water dumped on your head. By the way, armadillos, the Dasypus novum cinctus, when frightened, jump straight up. Doesn't make good defense when you're in the road. Okay, so again, the lag phase is, you can see that those are milliseconds. It is like less than a millisecond, basically. It's short. Contraction phase is roughly 30 to 40 milliseconds, thousandths of a second. And the relaxation period takes up to 100 milliseconds to finish it. So you go through the whole phase in basically a thousandth of a second. Okay, a tenth of a second. It's fast. To me, it's faster. All right, tetanus. When you get Clostridium tetani, the bacteria that causes tetanus, tetanus is when the muscles remain contracted with no relaxation. So the stimuli are so rapid, or the acetylcholinesterase is not there to break away the acetylcholine, and you stay flexed until, of course, the ATP runs out, and then you stop. So the tetanus, you can see in the picture here, the back muscles usually tighten up first. So you get that arching of the back. It's also called lockjaw because the masseter, the muscles in the mouth, would start, that shut the jaw, would start clenching. So you couldn't open your mouth to talk. So tetany is when the muscles under contraction so long, it just stays there. And then you fatigue. So fatigue, again, when the ATP runs out, and yeah, it's all over then. So tetanus kills you because the heart's also a muscle, and the heart tends to eventually lock up. All right, recruitment. Like I said, you can pick up a pencil, you can recruit a few hundred muscle fibers, muscle cells, or you can recruit a whole lot, but you gradually do it. You increase the number of motor units. A motor unit is the nerve innervating a certain number of muscle fibers, okay? So you can recruit a few to pick up something soft, light, or recruit a whole bunch to do a more stronger force. What is muscle tone? Those of you who work out, the body's constantly releasing enough acetylcholine, enough action potential to keep those muscles slightly contracted. So if you're tone, your muscles are not totally saggy. All right, these two are muy importante. Isotonic. Iso, same. So tone, same amount of tone, same amount of force, but the muscle moves. So it's a nice smooth, not a jerky movement, nice smooth movement. So you can pick that weight up nice and smoothly. That's an isotonic contraction. Diagram B, the person's lifting, but because the cable is bolted to the board, that muscle's flexing, but it's not moving. So metric means measurement, or movement, distance. So isometric contractions are when you're flexing really hard, but the arm's not moving. All right. And then, like I say, we learned in the lab, fast twitch versus slow twitch. The fast twitch is the white meat. The slow twitch is the dark meat. What makes the dark meat dark? Myoglobin. Myoglobin sounds like hemoglobin because it has the same sort of function. It holds oxygen. So when you're running and running and running, you can keep supplying oxygen to keep that ATP supply going. Now, the problem is the myoglobin tends to slow down that muscle so it doesn't twitch as fast. So distance runners, they build up a lot of dark meat, a lot of slow twitch muscle, so they can run and run and run. Fast twitch, people build up the white meat, the fast twitch muscle. Now, we were designed in certain ways when we come out that we're either higher propensity fast twitcher, higher propensity slow twitch. Usain Bolt from Jamaica, he's got a very high percentage of fast twitch muscle. He was just born that way. And he worked it out to where he became a world-class sprinter. You got people who are world-class marathon runners who were born with a lot of slow twitch muscle, so they could just keep going and going and going. But we can all work on building either one. Slow twitch is darker due to myoglobin. Go back and look at that for a second. You can pause this if you want. And then shivering. Remember, shivering, when it's cold, we shiver. When those muscles are going, you're releasing heat. You know that. You come in from PE class or from exercising, your face is flush. You're hot. Those superficial blood vessels have relaxed and your face gets red because you're trying to release heat. When it's cold, your superficial blood vessels constrict to try to keep that heat in so you get enough blood to feed your superficial aspects of your body, the integumentary system, but you don't lose a lot of heat that way. Same thing when you're cold, your muscles shiver to try to keep heat, build up heat so you can keep your body warm. And that's all.